Hello everyone. I am Dr. K. Vijay Chandra, Assistant Professor in the Department of Electronics and Instrumentation Engineering, VNR VIGNAN JYOTHI. Today I would like to discuss about analytical instrumentation, the introduction to analytical instrumentation. So analytical instrumentation is a basic course for fourth year BTech students, and usually this has a wide variety of applications. So analytical instrumentation, usually if you see the applications, you can see in pharmaceutical industry, similarly research and development, and similarly we have a lot of companies where it depends upon these analytical instruments. So usually coming to the biotechnology, also one of the field where these analytical instruments are used. So there are so many sophisticated analytical instruments are there, like spectrophotometer, infrared spectrophotometer, and nuclear magnetic resonance spectrophotometer, mass spectroscopy. All these are very sophisticated instruments, and the cost of these instruments are also very high. So it can range from some crores to several crores. So usually why these instruments are very important, if you ask any question, so I would answer that these instruments are mostly used for drug manufacturing. When in pharmaceutical industry, if you take, when they are manufacturing the drug, they have to rely on exact measurement. Otherwise what will happen, that measurement can go, if it is wrong, the property of the drug may get changed. So that is a problem. So that and that too, the drug quantity and quality is very essence. So if you take the quantity, usually we'll take in very milligrams or nanograms or micrograms. Sometimes nanograms also we should add that particular ingredient of the in the drug, because we know that usually the drug means there are several combination of the other drugs included to make a proper drug. For example, if you take the pharmaceutical, this usually Dolo 650 or paracetamol 650 tablet, 650 milligram is a one. Paracetamol is usually a chemical, but there are so many several ingredients are added into that in order to get the property, exact property, so that it can directly affect our illness and it can reduce. But if suppose the quantity of that particular ingredient, if it is slightly high or low, the property may get changed. So that's why in order to exactly measure the ingredient or component, we should rely on these analytical instruments. So far if you take that, several instruments are there, like spectrophotometer, infrared spectrophotometer, and so on. All these are used for different samples. Like we know different drugs are there. For example, take the liquid drugs are there, solid drugs are there, semi-solid are there, gaseous drugs are also there. For asthma patients, usually they'll take the this gaseous form of drug. Similarly, for infants, if you take, they will be relying on this mostly on the liquid type of drugs. So whatever it is, the drug, that quantity is very important, as well as the quality. Both can be measured with these analytical instruments. So today I would like to discuss the basic part of this analytical instrumentation. We'll see that introduction to this analytical instrumentation and these sophisticated instruments. Let me just say here, this is the analytical, usually if you take the definition, what is analytical instrumentation? So analytical instrumentation is the study of the separation, identification, and quantification of the chemical components of natural and artificial materials. So usually if you take analytical instrument, anything that could be anything, like a UV visible spectrophotometer or IR or flame photometer, anything that could be, it is a study of usually separation. Usually you can separate the sample. Suppose I told you that drug consists of so many ingredients, right? In that so many ingredients, you need to separate each component in order to identify the what is exact composition and what is the exact quantity of that particular separated separated element. So first of all, any analytical instrument, you need to separate that, and you need to identify it, and after that you need to quantify it, how much quantity it has, whether it is in milligrams, micrograms, or nanograms. You need to quantify it. First separate, and then identify, and then quantify. This is the major three things that any analytical instrument can be used for. And of a chemical components of natural and artificial materials, we know that in a periodic table, our periodic table consists of 118 elements. Each one has its own significance, and if you take, those are all, naturally naturally we can find those in chemicals, those elements, or some are artificial. By combining the two other elements, you can form the any artificial elements. Likewise, any artificial or natural, you are doing this. So, and usually there are several elements that elements are there in these analytical instruments. If you see here, first of all, this is the chemical information source. Chemical information source, suppose any drug is there, any chemical is there. So how do you want to extract those chemicals? By just igniting, whether you are igniting that chemical with the flame, or whether you are giving some electrical pulse to the sample, that chemical, or whether you are supplying any heat, or whether you are throwing any light onto the substance, like that you can do anything. So usually this chemical information source is how it is going to be ignited. That is what, once it is ignited, it releases some energy in the form of heat, or in the form of light, in the form of heat or light, or in the form of some electrical quantity, in the form of electrical quantity. So whatever it is, once it is ignited, once it is stimulated by this external energy, it will release this electrical quantity or heat or light. A proportional transducer you are connecting here. For example, if the heat is a liberated source from the chemical, what you do here, usually you will keep a transducers which is detects a heat indication, like thermometers, thermocouples, thermopoles, bolometers, or so on. Or if your energy source which was liberated by the chemical was light, you are depending on the photo transducers. Like photo transducers, or several types are there. Photo transducers, usually if you see, photodiodes, transistors, LDR, and similarly photovoltaic cells, photoconductive cells, photoemissive cells. There are several types are there. Or if your electrical source, suppose your chemical is liberating any electrical quantity, so you are relying on the usual transducers, that is voltage transducers, and so on. Usually electrical quantity is output of the output of any transducer, which is usually the definition is converting one form of the definition of a transducer is converting the energy, one form of energy to the other form. So usually electrical quantity you are converting, and that electrical quantity you are taking here, and you are applying a proper signal conditioning here. So because electrical quantity has numerous spurious noise signals, it consists, because the environment also has lot of noise signals, right? So what we'll do, usually we'll signal condition it properly, like by applying amplifiers, proper amplifiers, and chopping down to the normal level, and using the proper A to D converters, because if you convert from analog to digital, it is very difficult, the noise signal in order naturally any noise signal present in the environment is analog form. Suppose if you want to contaminate to our original signal, if it is analog form, the original signal, it is easily you can contaminate. But when you convert from analog to digital, we can you can make that noise free, and it is very difficult to interfere that noise signal into the digital signal. That is the reason why usually we convert into analog to digital signal, and from that we are going to display a proper display. So this is the usually elements of analytical instruments. So a chemical information source, and then you're converting a proper transducer, And these proper transducer is usually any sensor or any transducer, that physical form, it converts from physical form to the electrical form. And these electrical form you are directly giving to the signal conditioning circuit, and properly you are doing the signal conditioning, and then you are displaying. So this is the usual elements of the analytical instrument. And if you see the block diagram of analytical instruments, this is a block diagram of analytical instrument. And usually this is the sample, my sample. And say take the liquid sample, it could be the liquid sample. See here I am taking the example of the pharmaceutical drugs, anything that could be pharmaceutical drugs. So this is a liquid sample, or solid, or you can say liquid solid, or semi liquid, or semi solid, or gaseous sample, anything that could be. What you are doing is you are first igniting, or you are just stimulating this sample with the help of the following sources. See, these are the following sources. You can stimulus the sample with the help of light, or with the help of heat, or you can generate the current and throw onto the sample, or you can put onto the voltage onto the sample, so that it will get reacted with this stimulus signal, light, heat, and current and voltage. Usually we rely on this light signal. All analytical instruments so far I discussed, like UV visible and FTIR, like Fourier transform infrared spectroscopy, and so on, all are relied on this light source, whether it is a visible source, or ultraviolet source, or that could be infrared source, or anything. So whatever it is, you are throwing this proper source onto the sample, and the sample will react with this stimulus signal. And what it does, it takes the light, it takes the appropriate light. Suppose if I take the liquid samples, say some 10 ppm level of concentration of some liquid drug I have taken. Depending upon the concentration of the sample, this light will get absorbed by the sample, and remaining light will be emitted, or that is transmitted light. That transmitted light will send to the proper a photo detector, input transducer detector. That could be a photo detectors, photo transducers, because I have taken the light as a source here. That is the reason why I am selecting here a photo transducers. Anything that could be a photodiode or photo transducer, and LDR, anything. And your electrical signal will this light signal, the transmitted light signal from the sample source hit onto the detector, and from here it converts a proper photo transducer. It takes the light signal as an input, and gives the electrical signal as a output. This electrical signal you are sending to the proper signal processor, that is the proper amplifiers. You are amplifying that electrical signal, and you are digitizing that to avoid the noise signal. And finally you are projecting onto this meter, readout meter, like meter, plotter, or computer, anything that could be any. If you want to see the raw signal, electrical signal, you can also see the raw electrical signal or current signal. But after getting the electrical signal, usually our humans understand only physical signal, whereas the instruments understand only the electrical signal. And if you take this electrical signal, what we generally does, that you need to calibrate in terms of physical signal, and you are projecting onto the proper display to understand by the humans. So usually we does that. So this is a block diagram of this analytical instrument. Or suppose if you are not using the light as a source for the stimulus, to stimulus that sample, you can also generate a heat in the form of flame or something like that, and you can give the proper heat to the sample, so that the electrons jumps from lower energy state to the higher energy state by this liquid sample or solid sample. And after that, it after some time, 10 to the power of minus 8 seconds, again it releases some energy. That radiation is again captured by the proper transducer, and then the remaining prospects will be followed. And if you are not going with the light and heat, if you are selecting the current also, you can stimulus the signal, or voltage, anything. So that could be anything. Stimulus signal can be anything of this four elements: light, heat, or current, and voltage. So this is a general block diagram of analytical instrument. If you take any instrument, analytical instrument, that right now using in industries like Arabindo, Dr. Reddy's, and Mylon, all are depend upon upon these analytical instruments. Usually they have the source. See, for example, I told you that here is the light, right? Here is the light signal, right? I have taken the light signal. So what kind of light you are taking as an input source for the analytical instruments? So it depends upon this one. It depends upon this electromagnetic spectrum. See, it is if you see this electromagnetic spectrum, usually there are several lights or depends or several lights are involved in this electromagnetic spectrum. See, for example, see here, it starts from radio waves. See, this is the radio wave, and when you see this, this is the radio waves. So radio waves having the wavelength, this is the wavelength, 10 to the power of 3, and similarly microwaves has little bit wavelength is very less. Here it is shown. If you see here, this is the wavelength, right? So let me just draw this. So this is the any trough to trough or crust to crust. When you take the measurement, this is the wavelength, lambda. Usually you can represent that in the form of lambda, and usually this is radio waves. If you observe, it has a huge size. That wavelength would be very high, and usually whatever we are now depend on these radio signals only. For example, our mobile phones, if you take, we are relayed. How do we get the call? Because of these radio waves, electromagnetic radio waves, and which has a highest wavelength, maximum wavelength. Similarly, microwaves are there. These microwaves are also a little bit less wavelength. Wavelength is little bit less compared to the radio waves. But usually, what are the applications you can see in this microwave? Microwave ovens are the application. There you are using these microwave generators or microwave signals. And similarly, infrared is there. Infrared is a light source. Usually, it is not visible to the human eye. But infrared is a very crucial instrument which are used in drug manufacturing and pharmaceutical industries and R&D also. And this is the wavelength. If you observe, it is further less. See, 10 to the power of -3 to -6. If you observe, 10 to the power of -3 to 10 to the power of -6. And similarly, it goes here. The microwave is 10 and radio waves is 10 to the power of 3. And when you come to the visible range, this is the visible range. Usually, why here I indicated these different forms of spectrums is there. The color band is there because it is visible to the human eye. VJR spectrum. We are familiar with the VJR spectrum, right? So this is what the VJR spectrum. VJR spectrum. So violet, indigo, brown and green, yellow and orange and red. If you observe here, it starts from violet, indigo, blue, green and yellow and orange and red. Red has the highest wavelength, and which can start from 100 nanometers to 700 nanometers. And this is usually a human band, I mean a visible band, where our eyes are sensitive enough to watch this band. Whereas infrared, microwave, if you take, we don't see that. And similarly, X-rays and gamma rays also, we don't feel that, we don't see that. That is one. And again, if you see the spectrum, it is also further classified into ultraviolet A and ultraviolet B and ultraviolet C. So usually ultraviolet C are used for medical applications. For example, if you take for killing the bacteria. And similarly, in our general water purifiers in our home, there are one UV filter is there. That UV filter mostly it has the wavelength of 100 to 280 nanometer. Mostly our environmental bacteria or virus wavelength, that size of that bacteria would be around 100 to 280 nanometer in between. That is the reason why UVC exclusively used to kill that bacteria in our water filter. And similarly, for any bacterial killing applications in operation theaters, usually UV sterilizations are used. That UV sterilizations, which it kills that entire environment with that light, and it protects a lot in order to avoid any contamination to the human flesh when they are operating affecting the human flesh. And this is the ultraviolet C, and the UV ultraviolet B is there, ultraviolet A is there. Usually if you observe here, it is a burning, you can feel the burning sensation. It is ultraviolet B, which has the range of 280 nanometer to, okay, 320 nanometer. And similarly 320 to 400 nanometer aging, you can feel aging, which is a very danger for our DNA. Usually from the natural sun also we will get the ultraviolet region, where it is protected by our ozone layer. It will be filtered by our ozone layer, and the remaining light will be the visible light only will be fall onto the earth usually. But if there won't be any ozone layer, if you example if you take, what will happen, the entire light comes to our human bodies, or it projects onto the life which are present on earth, which kills, it can rip our DNA, so it can change the mutations of the DNA. That ultraviolet ray, that is the dangerous radiation effect we'll see. So we are lucky enough that we have the ozone layer where it is protecting, but at the same time ultraviolet C range we are usually used for so many applications. And we have the UV visible spectrophotometer, ultraviolet spectrophotometers, and similarly visible spectrophotometers are also there, we'll see that. And this is the action spectrum, see 322 650 nanometer, and this is absorption peak at 408 nanometer. We'll see that what it is that. So usually if you observe, this is a electromagnetic spectrum, and we have here this is a visible range. And similarly X-rays are there. X-rays, we are already familiar with these X-rays, right? X-rays are usually for bone density, or for any bone fracture, we will go for this X-rays. Okay, we are familiar with that, is also one device. Similarly gamma rays are there. There are certain spectrophotometers are there, exclusively for R&D purpose they are using, where gamma rays are used. That is nuclear rays. Gamma rays is nothing but nuclear rays. So if you take here this one, these rays, all the different electromagnetic radiations, that is radio waves and microwaves, infrared waves, and infrared and visible ray, visible and X-ray and gamma rays, all these are used in the source, as a light source in the spectrophotometer. See, for example, radio source if you are using, that could be a radio wave spectrophotometer, and simply microwave if you are using microwave spectrophotometer. Similarly if you are using infrared as a source in the spectrophotometer, that could be infrared spectrophotometer. Similarly X-ray if you take, so this could be the X-ray spectrophotometer. Similarly if you take the gamma ray spectrophotometer, and again in this gamma ray, further it is classified alpha, beta and gamma. We are using gamma here, but alpha, beta, it also comes under nuclear radiation. So that could be also alpha ray spectrophotometer or beta ray spectrophotometer, that could be anything. So whatever it is, but my point here it is, this electromagnetic source is very important in order to analyze a few samples. Usually if you take the radio waves, this visible and ultraviolet rays, all these are used for liquid drug manufacturing, liquid drug identification. So this is a electromagnetic spectrum, a very important spectrum for all which analytical instruments. So if you take the applications of analytical instruments, there are several applications are there. So what I have done here, I have just projected here few applications, like pharmaceutical industry, pharmaceutical industry if you take, for drug discovery, how you're gonna discover the as per if you take the quantification or quality of the drug, and similarly quality assurance and regulatory compliances, also you are depending in the pharmaceutical side. And similarly clinical studies, several drugs are day by day, if you observe, every day there new drugs are coming into our market. So how do they analyze? on these instruments only they'll analyze and they'll release to the market. So there are two things are there actually, a vivo application and a vitro application. So usually vitro means they will test with the instruments like analytical instruments before the drug comes into the market. And vivo side, if you take, the vivo means where it they test on the animals, so like mammals. So usually they test on the rats or monkeys. Those are the closest descendants of the humans. Usually rats or monkeys, they test on that, and after this gets succeeded, then they will release the drug into the market. All this comes under vivo application. And vitro applications are there where they test on the analytical instruments, and if they feel satisfied with that result, then they will keep that drug into the market. So these are the pharmaceutical industry applications. Similarly, biotechnology applications are also there, like proteomics and genomics applications are there, to understand the protein structure and genomic structure or DNA structure of that particular biological cell, with the help of these analytical instruments, because NMR exclusively used for protein analysis, nuclear magnetic resonance spectrophotometer, they exclusively used for protein analysis, and how the protein shells and layers consists of, so that they will do that modification, and they will test the drug with so many iterations, and then they'll release finally the drug into the market after intensive testing on this NMR instrument. Similarly, you have the microbiology applications like rapid microbiology, and similarly food safety. If you just depend upon, see here, food safety, there are several foods are coming into the market, like packed foods are there, processed foods are there. All those can be consist a lot of preservatives. So how the people knows that how much quantity does preservatives are there inside that food? So how do we know that? It completely with the help of these analytical instruments only, they will analyze and assess the preservatives, and they will test, and they will check the how much fat content, how much oil content, and how much salt, and how much energy that does it give. All this kind of stuff can be made with these analytical instruments, measures the analytical instruments, and then they'll put the sticker on the backside of the this cover or anything like that. That is the food safety. And environmental applications, if you see, pollution control, there are several gas that are there around us. See, pollution control, if you take, like carbon monoxide, hydrocarbon, sulfur dioxide, all these are the several potent gases, which are greenhouse gases, hydrocarbons and carbon monoxides and all. They will measure these in the pollution control also. Similarly, pesticide residue analysis, they will test the pesticides also before they release into the market. Similarly, there are a lot of laboratory automation is there, like sample preparation, sample analysis, and similarly characterization of the sample by these analytical instruments and so on. So similarly, LIMS also one more application in the laboratory automation. So these are the several applications of analytical instruments. So if you observe here, there are several applications are there where we're gonna study in detail about each and every instrument, analytical instrument, and we'll also understand the working principle, how it operates, and what is the signal condition, and how it works. All this this stuff we're gonna understand in the coming classes, and these are the usual applications of analytical instruments. Thank you very much.
Like, share, comment, and subscribe VNR Digital Learning Resource YouTube channel.